Welcome to the lecture on structure and function of different lipid classes 1. In this lecture, you will learn about the structure and function of fatty acids, triglycerides, cholesterol, and ketone bodies. Fatty acids play a crucial role as precursors for synthesizing various lipid molecules, such as triglycerides, phospholipids, and complex lipids. Fatty acids are organic molecules consisting of a hydrocarbon chain with a carboxyl group at one end. Hydrocarbon chain is the main part of the fatty acid and consists of a series of carbon atoms linked together by single bonds, saturated fatty acids, or containing one or more double bonds, unsaturated fatty acids. The chain can vary in length, and the presence and arrangement of double bonds influence the fatty acid's properties. Carboxyl group, COOH, is found at one end of the hydrocarbon chain. The carboxyl group consists of a carbon atom double bonded to an oxygen atom and single bonded to a hydroxyl group. This carboxyl group gives the molecule its acidic properties and distinguishes it as a fatty acid. The other end of the fatty acid chain is called the methyl end, CH3, which is a single carbon atom bonded to 3 hydrogen atoms. There are 3 common numbering systems for fatty acids: delta numbering, Greek lettering, and omega numbering. In delta numbering system, the carbon atoms in the hydrocarbon chain are numbered starting from the carboxyl carbon, which is designated as carbon 1. In the Greek lettering system, the carbon atoms in the hydrocarbon chain are labeled with Greek letters, starting from the second carbon in delta numbering is known as alpha, followed by beta, gamma, and epsilon. In the omega numbering, the carbon counting from the methyl end, omega end, of the hydrocarbon chain. For example, omega-3 signifies that the first double bond is at the third carbon from the methyl end. Fatty acids can be categorized as saturated or unsaturated. Saturated fatty acids are characterized by the absence of double bonds in their hydrocarbon chains, signifying that each carbon atom is fully saturated with hydrogen atoms. These fatty acids tend to be solid at room temperature. Examples include propionic acid, 3 carbons, butyric acid, 4 carbons, capric acid, 10 carbons, palmitic acid, 16 carbons, and stearic acid, 18 carbons. Unsaturated fatty acids are characterized by the presence of one or more double bonds in their hydrocarbon chains. Monounsaturated fatty acids have a single double bond, while polyunsaturated fatty acids possess 2 or more double bonds. Depending on the number and location of these double bonds, unsaturated fatty acids can exhibit a liquid state at room temperature. Examples of monounsaturated fatty acids include palmitoleic acid, 16 carbons, 1 double bond at position 9, and oleic acid, 18 carbons, 1 double bond at position 9. Examples of polyunsaturated fatty acids include linoleic acid, 18 carbons, 2 double bonds at position 9, 12, alpha-linolenic acid, 18 carbons, 3 double bonds at position 9, 12, 15, and arachidonic acid, 20 carbons, 4 double bonds at position 5, 8, 11, 14. Unsaturated fatty acids can exhibit geometrical isomerism at the double bond, leading to the formation of 2 distinct types, cis fats and trans fats. The terms cis and trans refer to the relative positions of the 2 hydrogen atoms situated next to the carbon atoms involved in the double bond. For cis fatty acids, the configuration involves the hydrogen atoms adjacent to the double bond being on the same side of the carbon chain. This results in a bent or kinked shape, influencing their physical state. Cis fats are commonly found in naturally occurring unsaturated fats, such as those present in vegetable oils. They are generally considered healthier than trans fats, and diets rich in cis fats, especially monounsaturated and polyunsaturated fats, are associated with cardiovascular benefits. On the other hand, trans fatty acids have a configuration where the hydrogen atoms adjacent to the double bond are on opposite sides of the carbon chain. This imparts a straighter structure to trans fats, similar to saturated fats, affecting their physical state. The majority of trans fats are industrially produced through the partial hydrogenation of vegetable oils. High intake of artificial trans fats has been linked to adverse health effects, including an increased risk of cardiovascular disease. Essential fatty acids are those that the human body cannot synthesize independently and must be acquired through the diet. There are two main types: omega-3 fatty acids, encompassing alpha-linolenic acid, ALA, eicosapentaenoic acid, EPA, and docosahexaenoic acid, DHA, found in fatty fish, flax seeds, chia seeds, and walnuts, exhibiting anti-inflammatory properties, and omega-6 fatty acids, including linoleic acid, LA, and arachidonic acid, AA, found in various vegetable oils, nuts, and seeds, demonstrating pro-inflammatory properties. Essential fatty acids play critical roles in physiological processes, influencing cell membrane structure, nervous system function, and the production of signaling molecules, eicosanoids. Non-essential fatty acids, in contrast, are synthesized by the body, and there is no strict dietary requirement for them. Common non-essential fatty acids, like saturated fats and monounsaturated fats, can be produced from dietary carbohydrates and other fatty acids. These fatty acids, although internally generated, still serve essential functions in biological processes such as energy storage, insulation, and cell structure. They can be obtained from both dietary sources and endogenous synthesis within the body. Fatty acids hold significant clinical implications across various aspects of health. Omega-3 fatty acids, particularly EPA and DHA, contribute to lowering triglyceride levels, mitigating inflammation, and enhancing overall heart health. Diets abundant in monounsaturated fats aid in balancing HDL and LDL cholesterol, fostering cardiovascular well-being. Omega-3 fatty acids exhibit anti-inflammatory properties, potentially offering benefits in conditions associated with chronic inflammation, such as rheumatoid arthritis and inflammatory bowel diseases. DHA, a specific type of omega-3 fatty acid, plays a crucial role in brain cell membranes, contributing to cognitive function and reducing the risk of neurodegenerative diseases. Both omega-6 and omega-3 fatty acids are essential for maintaining optimal skin health. Omega-3 fatty acids may possess anti-aging properties, contributing to overall longevity. Triacylglycerides, commonly referred to as triglycerides, represent a type of neutral fat crucial for serving as a primary storage form of energy within the body. Their structure is characterized by a glycerol backbone, consisting of a 3-carbon alcohol with a hydroxyl group attached to each carbon, to which 3 fatty acid chains are linked through ester bonds. There are two main types of triglycerides. Simple triglycerides, comprising 3 identical fatty acids attached to a glycerol molecule. Mixed triglycerides, encompassing a combination of different fatty acids linked to a glycerol molecule. Triglycerides perform several vital functions within the body. Triglycerides, the primary component of dietary fat stored in adipose tissue, serve as a highly concentrated form of energy. Adipose tissue, where triglycerides are stored, acts as an insulator, contributing to the maintenance of body temperature. Stored in adipose tissue around organs, triglycerides provide cushioning and protection, helping to prevent physical damage to vital organs. Triglycerides store fatty acids, which can be released into the bloodstream and utilized by cells for energy production. During fasting or periods of low carbohydrate intake, triglycerides are broken down into fatty acids and ketone bodies, offering an alternative energy source, especially for the brain and muscles. Steroids represent a distinct class of lipids, characterized by a unique 4-ring structure. Within this class, cholesterol stands as a sterol, belonging to a subgroup of steroids. The structure of cholesterol is defined by molecular formula, C27H45OH. Steroid nucleus, comprising 4 fused hydrocarbon rings, denoted as A, B, C, and D8 carbon, branched hydrocarbon chain, attached to the C17 position of ring D hydroxyl group, OH, present at C3 of ring A double bond, existing between carbon atoms C5 and C6 of ring B. Methyl groups, CH3, located at carbon positions C10 and C13. Additionally, cholesterol can form a cholesterol ester through esterification, where the hydroxyl group at C3 of ring A of cholesterol reacts with a fatty acid. This process increases the hydrophobicity of the structure, compared to free cholesterol. Cholesterol plays diverse and crucial roles in the body. It is an integral component of cell membranes, where cholesterol plays a vital role in modulating membrane fluidity and ensuring structural integrity. Serving as a precursor, cholesterol contributes to the synthesis of various steroid hormones. These include glucocorticoids, example, cortisol, mineralocorticoids, example, aldosterone, and sex hormones, example, androgens, testosterone, estrogens, and progestins. Cholesterol acts as a precursor for the synthesis of bile acids and bile salts, essential for emulsifying and aiding in the absorption of fats during the digestive process. Cholesterol has an insulating effect on nerve fibers, supporting efficient nerve conduction and ensuring proper signaling within the nervous system. The cholesterol derivative, 7-dehydrocholesterol, serves as a precursor for the synthesis of vitamin D3, cholecalciferol, in the skin. This vitamin is essential for calcium homeostasis and plays a critical role in maintaining bone health. Ketone bodies are water-soluble compounds that the liver produces from fatty acids during periods of low carbohydrate availability. There are 3 types of ketone bodies. Acetoacetate. This beta-keto acid has the chemical formula CH3COCH2COO and contains both a ketone functional group and a carboxylic acid group. Beta-hydroxybutyrate. With the chemical formula CH3COCH2COO, beta-hydroxybutyrate is a hydroxy acid featuring a hydroxyl group and a ketone functional group. Acetone, the simplest ketone, with the chemical formula CH3,2CO, is a volatile compound. It is exhaled, contributing to the characteristic acetone breath often associated with ketosis. Ketone bodies are generated in response to low dietary carbohydrate availability, occurring during situations such as fasting, prolonged exercise, or adherence to a low-carbohydrate/ketogenic diet. In low-carbohydrate states, the body intensifies the breakdown of fatty acids, initiating the synthesis of ketone bodies through a process known as ketogenesis, primarily taking place in the liver. These ketone bodies fulfill essential functions. Ketone bodies act as an alternative energy source, particularly for the brain and other tissues, in instances where glucose availability is limited. Ketone bodies play a crucial role in sparing muscle protein. They achieve this by reducing the necessity for gluconeogenesis, ultimately preserving lean body mass during periods of low carbohydrate availability. Thank you for your attention throughout the lecture on the structure and function of fatty acids, triglycerides, cholesterol, and ketone bodies.